Well, hello everyone. We'll get started here now. Thank you for coming. The name of this session, the title of this session is Emerging In Vitro Methods for Livestock Performance. My name is Michael Dame. I'm from the University of Michigan Translational Tissue Modeling Laboratory. The co-convener, Dr. Matthias Costa from Western Veterinary College of Medicine out of the University of Saskatchewan. He couldn't make it from Saskatchewan, or Germany rather, and where he's at another meeting. But he was instrumental in shaping the session and in inviting some excellent speakers. Our first speaker today, I'll introduce. Her name is Dr. Vanessa Hale. Dr. Hale received her doctorate of veterinary medicine and PhD at Purdue University. Prior to that, she received a master's in teaching at Western New Mexico State University while teaching high school in Crown Point, New Mexico. She completed a postdoctoral fellowship with the Mayo Clinic Microbiome Program in the Center for Individualized Medicine. She is currently an assistant professor in the Department of Veterinary Preventative Medicine in the Ohio State University College of Veterinary Medicine. Her research explores the role of gut and urine microbiome in human and animal diseases, how gut and urine microbes metabolize environmental chemicals, and how this impacts the host. She also works with a naturally occurring canine model of bladder cancer to reveal ways in which we can manipulate the microbiome to prevent or treat diseases like bladder cancer. Dr. Hale?
Good morning. Thank you. It is my pleasure to be here with you this morning and to tell you a little bit about my work. So you can see that today we're going to be covering next generation sequencing and OG approaches to studying the microbiome. Now, I asked my husband in the hotel this morning, what does OG stand for? And he said, I don't know, Orange Grove. So just in case you're also not quite sure what OG stands for, it is slang for original gangster, meaning original or old school, but said with much respect. So we're going to be talking about microbial culture, but we're also going to be talking about microbial sequencing today. And let's see, we'll also talk about what is the urobiome, what are challenges that impact the study of the urobiome, and how do we study the urobiome? And this is really to get at these kinds of body sites that are low microbial biomass but high host biomass, and how do we think about them, whether it's in, for me, the context of things like bladder cancer, or whether it's in a context of something like production and performance. So, as you may already be very familiar with, the microbiome is the collection of archaea, viruses, bacteria, microbial eukaryotes on and in the body. So that's in us and that's in animals. And what you may also note is that in blue on your right side, that's the number of microbial genes in roughly a human host or mammalian host body. And down in orange on that right toe, that's the number of human genes. So you've got microbial genes to very, very few human genes. And so trying to understand what those microbes are capable of doing and the functions they may be carrying out on and in us becomes really important to understanding things related to health and performance. We already know, particularly in relation to gut microbes, but also in other body sites, that microbes play a key role in immune development, in nutrition, in behavior, in fitness, and in production. We know that microbes play a role in disease susceptibility, and they can help us predict prognosis and response to therapy. So I don't specifically work in performance and production, but there was just a recent Animal Microbiome Journal issue entirely dedicated to performance, optimal livestock microbiome performance. And so some of the topics that were highlighted within this issue were things like understanding resistance to Salmonella typhimurium using cecal microbiomes in poultry, looking at the weaning process in goats and giving them rumen fluid as a way to promote that weaning process, understanding feed efficiency in dairy cows, also using AI in the microbiome, looking at the skin microbiota in aquaculture, so in fish, trying to understand whether or not UV treatment would be beneficial in disease settings with aquaculture. And then There's also been work on fecal and vaginal microbiomes in beef heifers. And now this vaginal microbiome is kind of an area that's getting closer to an area that I work in, which is really these low microbial biomass, high host biomass microbiomes. And so a lot of what I'm talking about today is really talking about how do we study these particular types of microbiomes. So the urobiome is the microbiota of the urinary tract. And one of the questions that really drives me is trying to understand, does the urobiome play a role in bladder cancer? Now, I'm a veterinarian, I work in a college of veterinary medicine, and I ask this question in animal model species, pet dogs. So pet dogs have a bladder cancer that looks very, very, very similar to human bladder cancer. We know a lot about colon cancer and microbes. We know very little about bladder cancer and microbes. And that's because the challenges that we've faced in studying the urobiome. It is low microbial biomass, which means there's a high risk for introducing microbial DNA or contaminants in the extraction and sequencing process. It can also be potentially high host biomass. So in inflammatory conditions, things like bladder cancer where you may have blood cells or ulcerations, you may be shedding a lot more host cells or epithelial cells into the urine. That's a lot of host biomass that you then need to contend with in sequencing data. And so I'll talk to you a little bit about some studies that my lab has been doing to try to overcome some of these challenges. So first, I'll give you a little introduction to the urinary tract. So when we talk about the urinary tract, we're talking about from the kidneys to the ureters to the bladder to the urethra. So that's the full urinary tract. And the urinary tract looks very different than many other body sites. So here, MD means microbial density, and O2 is your oxygen tension. So we've done a lot of work in the gut microbiome, and we know that the gut can have a very low pH. It can be essentially anaerobic or fully anaerobic in the colon, and it can have a very high microbial density. The urinary tract can have a varying pH, but closer to neutral, has an oxygen tension somewhere between that of the colon and the mouth, so microaerophilic is typically what we think of it as, and it has a low microbial density. And so these are some of the aspects of this habitat that we have to try to understand when we want to study the microbial communities there. So how do we do that? Well, one of the first studies we did is we took 14 healthy adult dogs and we collected urine as well as some other samples from them. So we collected perineal and genital swabs as well as fecal samples from them at 12 different time points. The first 3 time points were morning and afternoon of day 1, day 2, and day 3. And this is really just to try to understand how changeable or stable is the urine microbiome. We don't even know if you keep the same microbes there over and over again or if it's new every time. Because if you think about the bladder, you're constantly taking in fluid and then releasing fluid. And so do you just have a totally new composition every couple hours? And so our first time points were trying to addressing differences by hours. And then we also collected 1 week apart, so week 1, week 2, week 3, and week 4. And then we collected month 2 and month 3 time points. So really trying to understand differences between hours, days, weeks, and months, and seeing if we have some stability there or just total randomness. And first, we used culture. So we used standard urine culture and then MALDI-TOF for identification. And this was largely driven by Dr. Andrew McGlynn, who recently graduated as a veterinarian and is now pursuing a residency. So some of the advantages of culture, when we culture urine, we know that we're obtaining viable microbes. And we can also get full genome sequencing of those isolates and to try to identify what functions those microbes have. The disadvantages is we're not always great at capturing everything in culture. So we can miss a lot of what is truly there and what is truly viable based on our standard urine culturing techniques. And I'll talk about that a little bit more in a minute. So what did we culture in healthy dog urine? Well, if your brain is hung up on a different question first, let's talk about that. So in case you were taught in a medical school or vet school, which still teaches this concept, which is urine is sterile, let's talk about that. Urine was thought to be sterile for a very long time. In fact, the original Human Microbiome Project, which launched in 2007, didn't include the bladder as a microbiome site because it was thought to be sterile. But there's been a lot of growing emerging evidence to suggest that there is in fact a healthy urine bacterial community that is present across individuals. So this comes in the form of culture-positive asymptomatic bacteria. So you can take someone who has no signs or an animal who has no urinary tract signs, and you can culture their urine, and you will get urine isolates, urine microbial isolates. We also see culture-negative symptomatic UTIs. So this is particularly common in the setting of recurrent UTIs, so whether that's in women or whether that's in female dogs, individuals who present over and over again with signs of urinary tract infection, and you send them to culture and you're like, I've got nothing, I don't know if I can give you antibiotics. In fact, if we pursue alternate culturing techniques, we can often find disease-causing agents, so uropathogens that are causing disease but don't get picked up in standard urine culture. When we used enhanced quantitative urine culture, which I'll tell you more about in just a minute, and next-generation sequencing, we find that there's bacteria in over 90% of individuals. And this is urine that's collected transurethral or using suprapubic aspirates. In dogs and cats, that's something called cystocentesis, so you put a needle directly into the bladder to obtain urine. In humans, that's called suprapubic aspirates. So these are very clean samples. These aren't just midstream voided samples. And these are coming from healthy asymptomatic individuals. So we are quite convinced that there is a urine microbial community in healthy individuals and that it's probably doing something there. And what I want to understand is, what is it doing there? So as I mentioned, we use standard urine culture, but something that we're expanding into is this new EQUIC, or expanded spectrum quantitative urine culture. So standard urine culture, so if you were to go to your doctor today and present with urinary tract symptoms, your urine would likely be collected, and 1 microliter of it would be plated onto blood agar, so BAP, or McConkey agar, and it would be incubated aerobically at 35 degrees Celsius for 24 hours. And if nothing showed up, they'd say, you're clean. Expanded spectrum uses 5 different types of media and 7 different types of culture or incubation conditions. So you've also got a chocolate agar, a colistin nalidixic acid agar, a blood anaerobe agar, and then you're looking at aerobic, anaerobic, and microaerophilic gas mixtures. And this has truly expanded the number of microbes that we're able to capture in urines, both healthy as well as urines from individuals who are presenting with urinary tract symptoms. So standard urine culture missed 67% of the uropathogens that EQUIC has been able to detect in various studies. So it really improves recovery of urine microbial isolates. And if this is a space you work in, or if you work in similar low biomass sites, considering expanding your culturing methods may be really valuable to you. So now we know there's microbes there, but what are they doing? Well, we have a pretty good idea that these microbes are doing a lot of similar things as the microbes in the gut. So we think they're probably producing neurotransmitters, they're outcompeting pathogens, they're regulating epithelial junctions, and producing things like antimicrobial compounds. They prime the immune defenses and promote development of the urinary tract and block pathogen access to the uroepithelium. So they're doing a lot of really important roles for us. And to try to understand, where are those microbes coming from? So it's a low biomass environment. Why are they there? How are they getting there? Well, let's see, I don't have a way to show you this. You're looking at the urinary bladder in red there, and in yellow and green you see the presence of microbes. Now this is human female anatomy. So right next to the urinary bladder you see the vagina going up to the uterus. And so there's a lot of anatomical proximity and sharing of potential microbes between vagina and bladder. And we know that that is the case, but we also know that some of these things that we're detecting in the vagina, like lactobacilli, can actually live and thrive and colonize in the bladder as well. They're not just vaginal microbes. We also know, particularly in females, based on anatomy, that you can see there in purple, you've got anus and rectum, and you've got gut microbiota. So that's a really high microbial density source of microbes in a really close proximity to the urethra. So you have some potential for microbial sharing there where you get ascent up the urethra into the bladder of potential gut microbes. Now, there's also some more question about, are there microbes that can be in the blood that can then also make it to the bladder through the blood? So certainly there's a presence of metabolites, but can we track potential microbes through blood to kidney to bladder? And that's something that continues to be worked out. So because I work in dogs, here's a quick review of dog anatomy. So on the left, you have males, and you can see just like in human males, you've got that opening to the rectum up near the tail, and the urethra and penis that are distant in proximity, as compared to the female, where that rectal opening is in the same place in the male and female, but then you also have the uterus and the bladder openings close by. So what did we culture in those healthy dogs? Well, here, the important thing is not to see exactly what all of those small words say, but what you're seeing in the rows across are individual dogs, and what you're seeing in the columns are individual time points. So remember, these time points are hours, days, and weeks apart. And what you can see is sometimes there's a dog where you see blue almost all the way across, or purple almost all the way across. These are animals where we cultured almost the same microbes at every single time point. But you can also see a handful of places where something red emerges one time, and we culture it one time, and then we don't culture it again. So we see there are some consistent taxa over time in urine cultures of some dogs. And this is based on culture. And this is my dog, and incidentally, we also did antimicrobial resistance profiles, and she's full of it, so that's fun to know. So then we also did 16S rRNA sequencing, where we sequenced urine, stool, genital swabs, and perineal swabs. Now, some advantages and disadvantages here. 16S can be more amenable to high host biomass. So you're targeting bacterial sequencing, or bacterial and archaeal. So you can get away with having higher host biomass, but still getting a really good representation of the microbial community. There's been some more recent work suggesting that high host biomass can still potentially interfere or bias 16S samples. So there's a little bit of plus and minus there. 16S is inexpensive, and it's relatively simple analysis, but it doesn't allow you to discriminate live versus dead. So you can say, hey, I see 300 taxa there, but are they all alive? You have no idea. Are they functioning? You can't assess function from 16S. So some pluses and minuses there, but often a good place to start in these low biomass body sites. So here what you can see in red, every dot represents an individual microbial community. And in red, those are fecal communities. In yellow, those are urine communities. So these are all from the same dogs, but you can see fecal and urine microbial communities are quite different. In blue, we have perineal swabs. The perineal area is the fur between, or the area between the rectum and the urethral opening. And so in this case, it is skin-based. And so you can see there's some shared microbes, and then some that are more similar to fecal, and then some that are just in the middle. The green and the purple are genital swabs, so preputial or vulvar. And you can see that urine and genital swabs do tend to share a lot more microbes as compared to fecal. So where are these microbes coming from, or where are they being shared between? The other thing that we observed that was quite interesting is over these 12 time points, you can see some of these dogs maintained a really stable urine microbial community, and some of them were more variable. So if you look down in the lower right-hand corner, you see all those pink triangles. That's all one dog, and that's over 3 months. And you can see that those microbial communities are really clustering, so they're very similar over time. There's other dogs like green circles where it's a little bit more variable. So there's still a cloud of similar microbial community, but it's more variable than the pink triangle dog. And so this told us that urobiome composition can be somewhat consistent, and that depends a little bit by dog. And this is valuable for us to know, because despite that vastly changing environment, it can change in pH, it can change in concentration and metabolites, it will still have a relatively consistent microbial composition. So one of the next things we wanted to try to understand is, does urine volume matter? This is a super basic question, but when we look at the human literature on this, they're reporting anywhere between the use of 0.5 mils to 50 mils of urine samples. And we have no idea if this matters or not. And importantly, if we're working in either conditions, so poor emptying of the bladder or frequent emptying of the bladder, or we're working in pediatric individuals, or we're working in species where there's no way we're going to get 50 mils of urine, so maybe we're working in kittens. One thing we wanted to understand is there some minimum threshold of urine that we can or should be using? Now, of course, this is caveated by we used dogs and we used free catch, and so if we do it in any other species, we got to figure that out. The other reason that was driving this is I would love to move into a rodent model to answer some mechanistic questions about the urobiome, but can I get away with 200 microliters is really kind of one of the questions I wanted an answer to. So this is Zach Lewis. He is about to graduate with his master's, and he did this study where using urine from the dogs from the previous study, we collected leftover urine from those dogs. It was 5 of them, and fractionated it into 6 volumes, going from 0.1 mils all the way up to 5 mils. Now, this isn't a huge amount, but we wanted to try to understand on this low end, can we get accurate representations of the urobiome? And so what he found was as the urine volume increased, the number of unique amplicon sequence variants, so here on the y-axis is amplicon sequence variants, that number increased. Amplicon sequence variants are really just a way of capturing microbial diversity. So in other words, we saw greater or captured greater microbial diversity as our urine volume increased, which is really suggesting we're able to capture more rare species as our urine volume increases. This is kind of an intuitive understanding, but it really helped us see that these small volumes, so less than 1 mil, really didn't allow us to accurately capture that urine microbial community. The other thing we looked at is we use a lot of negative controls. Every time we extract and sequence, we generate samples that have no sample and use them as extraction controls. We use swab controls, we use reagent controls. And when we look in those controls, we often see microbial signatures, but they are signatures of reagent ohm or kit ohm. And so we said, do we see more of these contaminating sequences depending on urine volume? And the answer was yes. Now, this wasn't significant, but you can see here what suggests a biological trend toward as you have higher urine volume, you're seeing fewer of these artifactual contaminant reads. And so it's really suggesting that the more urine you have, the better off you are at capturing the true microbial community and less likely to get this spurious or artifactual communities. So then when we looked at microbial composition, again, every figure or every dot here represents an individual urine microbial community, but the shapes represent dogs. So you can see the triangles are clustered together, the circles are clustered together. Dog generally overwhelmed any signature we saw by sample volume. So that was a good thing to know. But that said, when we look at an individual dog, so this is one dog, but if we look at, you can look at every single dog in the study and you see the same pattern. The dark colors or the darker colors represent higher urine volumes. The lighter colors are lower urine volumes. And what you can see immediately is those dark points, so 1 mil, 3 mils, 5 mils, they really cluster together. In other words, they're kind of reproducible in the microbial community that you're seeing. Those very light colors, they're all over the place. So you're seeing totally different microbial communities, probably some stochastic rare things that are showing up in each of those samples. And so it's not until you get to a higher volume that you can really get a reproducible urine microbial community. So that was valuable for us to know. So to answer our question, does urine sample volume matter? The answer was yes. So conceptually, if you look at this cup of urine, if you cut the amount of urine that you're using, this pink and this purple taxa, you may lose these rare taxa. And so what we determined was greater than 1 mil urine samples were optimal for consistent representative urine microbial community profiles. And this was by 16S rRNA sequencing. So now that we knew that, we wanted to tackle the other major challenge that we find in urobiome profiling, and that is high host biomass. So there's a lot of different methods already out there for trying to remove host DNA or deplete host DNA. So what we did was take urine from 7 healthy dogs and use 6 different extraction methods. Now, one of those methods did not include host depletion, but 5 of them included some step that involved host depletion. So separating out host DNA from microbial DNA and seeing if we could improve our sequencing of the microbial community. We also spiked these samples with additional canine cells at a biologically relevant level. So in speaking with our pathologist, one of our canine pathologists, we understand that about 75,000 cells per milliliter of urine is a normal read for a dog. So that's equivalent to like 1 or 2 host cells detected in a urinalysis profile. And so we spiked it at that level to really try to understand, are we effectively removing host cells with these host depletion methods? And then we performed shotgun metagenomics on this. And this is really where I wanted to be able to get to. So I want to understand functions of these microbes. So a metagenome is where we take these microbes and they have their own genomes. And then on the sequencer, we break up those sequences into short little fragments, and those are the filtered reads. And then we assemble them into contigs. So we take those short little reads, maybe 150 to 250 base pairs, and we put them together into contigs that belong together. And this is based on enough depth of sequencing that there's overlap that we can reliably assemble these contigs together. And once we have those contigs, then we can answer questions that are functional. So we can say, hey, are there antimicrobial resistance genes present in this dog, or in this condition or this condition? Are there the ability to degrade mucin in this dog or this dog? But the thing that's really powerful is if I can take those contigs and assemble them into full genomes, so I can say, here's these functions, and they are in this specific microbial strain, that's super valuable. And so that is generating metagenome assembled genomes. And that's the point that I wanted to try to understand. Can we do that in urine samples? They're low biomass, and we're going to have to sequence deeply in order to get enough reads to do that. So advantages of doing shotgun metagenomics, you're going to get everything in that sample. You're going to get bacteria, viruses, plasmids, microbial eukaryotes. You're going to get species and strain level resolution, and you're going to be able to get functional information. But the disadvantages are there's still no live or dead discrimination, and it is more expensive and a more complex analysis. Contamination by host DNA can also overwhelm. So if you have a ton of host DNA in there, you're going to get 90% host reads and 10% bacterial reads. And if that's really what your interest is, you're going to have to sequence really deeply to try to understand that 10% that is of your interest area. So when we looked at different extraction methods for depleting host DNA out of urine samples, we found on the left-hand side, for total DNA, the bacteremia, the Chi-Amp bacteremia kit was very effective at getting a lot of total DNA. Now, total DNA includes host and bacteria. When we looked specifically at bacterial DNA, what we saw was that DNA microbiome was actually more effective at getting bacterial DNA and depleting host DNA. So now we know how much DNA is there and how much of it is bacterial. And you can see, look at these y-axes, especially for bacterial DNA. This is not a lot of DNA that we have to work with. And so as proof of concept, we wanted to try to understand what can we do with this DNA. And so we profiled urine microbial diversity and composition. And again, DNA microbiome emerged as a kit that was particularly effective in capturing microbial diversity. So on the y-axis, you see Shannon entropy. That's a measure of microbial diversity that's weighted by abundance. And what you can see is that DNA microbiome recovered the greatest microbial diversity as compared to several other methods that we tested. But again, individual, or dog in this case, overwhelmed differences that we saw by extraction methods. So you can see here that kind of the pink samples are clustering together, the green samples are clustering together. Each color is an individual dog, and so even if we extracted it using different methods, largely we still got signatures by dog. So that was a valuable thing for us to know too. And then we attempted metagenome assembled genomes from urine. Now remember, it's 7 dogs, we're really testing extraction methods, but we really wanted to know, can we even get them? And the answer was yes. Now, we did not get a lot. We got 16 different metagenome assembled genomes, and you can see here, DNA microbiome, we were able to assemble more MAGs than all of the other methods. Now, these taxa are urine-related taxa, urine-derived taxa, particularly in dogs. So these seem to be legitimate taxa that we're looking at. And once we have a MAG, in other words, we have an entire microbial genome that never came from culture, that just came from sequencing, we're able to do the cool stuff. So we can now look at any functional pathway that you want to look at. Here's just an example of core metabolism. And we can look in each one of those MAGs and say, what kinds of core metabolism pathways do you have? And then look at those abundances. And that is a huge, powerful thing that we want to be able to do in these urine samples. So our key takeaways from this, in samples with high host biomass, the Kaya-AMP DNA microbiome kit yielded greatest bacterial DNA concentrations, highest microbial diversity, greatest number of MAGs, and it effectively depleted host DNA. In samples with low host biomass, our bacteremia kit was quite effective. So kind of thinking about what samples you're working with and if you need a lot of host depletion, how that might look. So conceptually, if you look at our sample in the middle, we call this our true sample. So the big pink circles, those are host cells, and the squiggly and the small pink bars, those are bacteria. The bacteremia kit is able to effectively capture a microbial community without depleting host DNA. So there's no steps in that kit to deplete host DNA. If you look to the right, this is what we hope and a really effective host depletion kit would do. It would just get rid of the host cells, but really capture the true representation of the microbial community without biasing it. And we saw this most effectively with DNA microbiome. On the other side, you see we had several kits that were able to get rid of host cells, but they did bias the microbial community, so we don't see those pink cells in there. And those are less ideal for us. So circling back to the very beginning of this talk, I mentioned that my driving question is, does the urobiome play a role in bladder cancer? Well, now we have urine MAGs, and we can answer some of the questions that I'm really interested in answering. So one of those questions is, environmental chemical exposures are associated with bladder cancer. And we know this from a host of epidemiological data in dogs and in humans, things like tobacco smoke, things like polycyclic aromatic hydrocarbons, these things that are produced through combustion processes and industrial emissions. What we don't know is, can microbes metabolize these chemicals? We know these chemicals, you get exposed to them and they get filtered into your bladder. What are microbes doing with them? We know there's microbes there, and we know they're sitting there with these chemicals. Can they metabolize these chemicals? Well, based on the MAGs from this study, the answer was yes. We found both full and partial pathways across multiple microbes for long-chain alkane degradation. So we don't know what this means. We don't know, are they detoxifying it for us? Are they generating secondary metabolites that are more toxic? We still don't know a lot of questions about this, but the fact that we now know these microbes inside of us have the ability to degrade these chemicals that can also be inside of us is really important for us to understand in something like bladder cancer. And so an example here, Staphylococcus pseudointermedius has this LAD-A, which is this long-chain alkane dehydrogenase, and we see a pathway in this Staphylococcus pseudointermedius where we can see the degradation steps occurring. Now, the other interesting piece of this is everything that we did initially was in healthy dogs. We also tried this in dogs with bladder cancer and said, can we generate MAGs and can we look at their functions? And we were able to generate an Enterococcus faecalis MAG from the urine of dogs with and without bladder cancer. So the thing is, they both have, whether they're treatment or control group, they both have Enterococcus faecalis. But in the dogs with bladder cancer, that Enterococcus faecalis contained genes for polycyclic aromatic hydrocarbon degradation. That's, again, an industrial environmental chemical. But in healthy dogs, the Enterococcus faecalis did not contain those same degradation genes. So is that a difference due to exposure? Is that a difference contributing to disease process? We have no idea, but it becomes really important in our understanding of bladder cancer, which has been linked to PAH exposure. This also really points out that strain differences are critical for us to understand. So, we could do 16S all day and say, hey, Enterococcus faecalis is in everybody, but if we don't understand the functional differences between bladder cancer and healthy, that doesn't help us get at what might be a really critical piece of biology for us. So I've highlighted already the ways we study the microbiome. So there's culture, which gives us the viable microbes, but some microbes are challenging to culture. There's 16S sequencing, which is targeted, but doesn't allow us to discriminate live and dead. And there's shotgun metagenomic sequencing, which allows us to get full functional potential, but cost and analysis becomes a little bit more expensive and more complex. So our final takeaways here: healthy urine contains a diverse microbial community. Urine samples of greater than 1 ml really produced our most consistent urobiome profiles in healthy dogs. Inter-individual differences overwhelmed differences due to DNA extraction method. And there are host depletion methods that were effective in the face of high host biomass. We, as proof of principle, determined that MAG assembly is feasible, but it's limited in low biomass urine samples. So again, we had 16 MAGs that we assembled. That's not very much, but maximizing urine volume would likely improve MAG recovery. So if you are working with a condition or a species where you can get 50 or 500 microliters, you should do that. Milliliters. And in those samples, even without MAG generation, you can use gene-based queries. So you can say how many antimicrobial resistance genes are present in these contigs. You don't have to assemble the MAGs for that. But it will be much more challenging to link the function to the specific taxa. And as we saw with that Enterococcus faecalis example, that could be really powerful. And finally, urine-derived MAGs revealed evidence for environmental chemical degradation, and that has a lot of relevance for host health as well as bladder cancer, and likely relevance for things like performance as well. So this is my team. We just went ziplining very bravely, I feel. And I'm very grateful to a really wonderful team, as well as funding and collaborators that have supported all of this work.
Thank you very much, Vanessa. That was excellent. Very, very interesting story. Could we have questions, please?
Yeah, very, very, very interesting study. When you get the stable microbial populations, I have a couple of experimental questions. First off, was that male and female dogs, or was it just female dogs?
We did not see a difference in that stability by sex, but that's a good question. It was a total of 14 dogs, 7 males, 7 females, so that would be a little bit harder for us to detect based on that power. We did test for it and didn't see it, but I understand the heart of your question, and I think if we had a bigger sample size, we might be able to address it even better.
Yeah, that would be interesting. And experimentally, when you collected the urine samples, did you do analysis of the flora, or did you freeze them down all together?
We cultured immediately, and then we froze, and then did a full batch of extractions and sequencing together to limit that variability that could come in with extraction and sequencing batches.
Then one quick comment for your PAH studies. The induction of genes, I would suspect, went through the aerial hydrocarbon receptor. And knocking that out or not may tweak out if involved. Explore that a little bit more, or that's a future studies?
That's an open question. We see several different possibilities for the way these microbes might be metabolizing it. So there's some bacterial CYP genes. There's also some pathways, and we learn this from soil and environmental microbes, where there's a lot more work done on things like PAH degradation. There's AHR-related pathways, but I think there may be additional pathways that we will continue to try to discover, and some of that we're working on now in vitro, so like actually exposing microbes to PAHs and saying, what are you doing and how are you doing it?
Let you go, but any suspicions as to the non-aerial hydrocarbon?
So I think the answer is yes, but I don't know that. I think there's both aerobic and anaerobic methods for PAH metabolism. But again, I don't know if that's happening, and I don't know if that's happening in hosts. But if you know more about this, I would love to talk about this and kind of keep an eye on what we should be looking for and genes we should be looking up and testing for. Thank you for those questions.
In humans, urinary tract infections increase with menopause. Is there a difference between neutered and not neutered female dogs?
So you said in humans, urinary tract infections increase with metabolism?
Menopause.
Oh, with menopause. So it is more common in females. Does it change with spay or neuter status? I don't know the answer to that. There's probably an answer out there, and that's a good question. So many of our animals are spayed, and in this specific study, we focused only on spayed animals, but I can look that up and see if there is already known work on that, because I suspect that there may be a difference there.
Thank you.
Thank you very much, Vanessa. It was excellent. Thank you. Well, our final speaker today is Dr. Mariana Meneguzzi, and she is a native of Brazil, where she obtained her Doctor of Veterinary Medicine in 2018. In 2020, she graduated from the master's degree from the University of Minnesota under the guidance of Dr. Matthias Costa, our co-convener. Currently, she is pursuing her PhD under the supervision of Dr. Noel Joez and Dr. Montserrat Torre-Morel at the University of Minnesota. Her research focuses on the development and application of a novel swine viromic assay that combines beta enrichment with long-read, real-time sequencing to better understand swine viral co-infections. The title of her talk, Exploring Viral Co-infections Using the Televirus Workflow.
Thank you so much for the introduction. Thank you for everyone. So as was said, my name is Mariana, and I'm currently doing my PhD at the University of Minnesota. And I'm very happy to be here presenting my project, which consists in the development of a workflow that is called test virus. So test virus, we have several goals for the workflow, but one of them is to better improve the way you do virus surveillance, but also the way we study viral co-infections. So to start a little bit, when we think about virus and virus co-infections, those are very well studied. We have a lot of research in the literature, but and those co-infections seems to be important for clinical outcomes. So it seems that the greater the number of the virus that we have co-infecting the animal or the humans, we have increasing clinical signs. However, we do have some gaps in the literature, and this is mainly because we have very few large epidemiological studies in commercial livestock, and most of the studies only have 2 or 3 viruses, and virus variants are not typically characterized. And the main reason for this gap is methodological. And I'm actually very happy the 2 other speakers, they did talk about virus in swine, and also the problem we have with clinical or field samples, which consist in very low amount of the input and a lot of host nucleic acid. And that's one of the reasons. So what we have about when we use clinical or field samples, we must encounter this issue, which is very few proportions of viral nucleic acid and greater proportions of other nucleic acid. And this makes our life very, very hard when we think about sequencing those samples. What we currently do to actually sequence or isolate the virus of interest is to do in vitro in culture the virus prior to sequence, or we do PCR amplicon sequencing. And those techniques, those methods, they have a lot of advantages, but they also have disadvantages, such as we can alter the virus population structure when we do that. Sometimes we are not able to detect novel variants or to recover the full-length genome. There is an ability to capture the true genomic diversity within the viral population. And if we are interested in isolate several viruses, we must need to do several workflows, and this actually leads to a multi-day turnaround time. So because of those advantages from the current methods is where actually came our idea to combine a method called probe capture and enrichment method, followed by long-read sequencing. And then is when born TS virus. TS virus workflow stands for target enrich long-read sequencing of virus, and our goal was to generate this workflow that would be able to reconstruct unbiased, well-characterized whole viral genome sequences. Here is our overview of the workflow. I broke down in 4 main steps, and I go through them a little bit. So the first one is the probe design. So we need to design the probes, and in our case, for my project, we designed a probe set that was able to target a total of 40 swine virus. So first of all, we did a list of 40 swine virus that we thought had economic importance for the swine industry, but also it can have an emergent potential or zoonotic potential. So we had the list, and then we download all we download the full-length genomes that were available in NCBI for those viruses, and then we design debates using the SIOT algorithm. The SIOT algorithm was developed by Dr. Jarno Lanko, and is already published. The probe comprised a total of 16 full-length genomes, and the length of the probes are 120 base pairs, and the probes cover 100% of the genome. So after we have the probe set is designed, we can actually go to the lab and do the second step, which consists in the probe capture and enrichment method. So for that, the concept is that we're gonna have the sample, the field or clinical sample. We're gonna have our probes that were just designed, and what happens is the probe is gonna be added to the field sample and gonna be able to target the virus of interest. After that, we are able to do some washing, and then after the wash, we can be able to amplify this target sequence, target nucleic acid from the virus of interest. And then the whole idea is that then we're gonna have much more virus nucleic acid compared to the actually host or other nucleic acid. And then after we do the probe capture and enrichment method, we follow by long-read sequencing. And we choose to use an Oxford Nanopore technology platform. We use a Minion MK1C, which is a portable device. It generates long reads, which is another advantage of using third-generation sequencing, real-time, and is fast to actually sequence. So after we have the sequence finished, we can collect the data, and we do some bioinformatics. So for the bioinformatics, I not go very deep. We can, if you're curious, you can actually check the GitHub page because it was already published for TS virus. We're gonna have the rates, and then we can classify those rates in host rates. So the rates that align to the host, in our case, the swine, And then we also can classify the rates that were generated in non-target rates, so the rates that actually align to the virus of interest, the genome of the virus of interest. The bioinformatics pipeline was developed, I just forgot to mention, was developed in collaboration with computer scientists from University of Florida, Dr. Christina Bolsher and Jonathan Bravo. So here is just another overview with more details of the TS virus workflow. So we start with the field sample, we do RNA extraction, cDNA sense, library preparation, then we sequence. So the library preparation consists also of the bait and the probe capture enrichment method, then long-read sequencing, and then we are able to do, we do the bioinformatic pipeline. So this methodology and the combination of bait capture enrichment method with long-read sequencing was previously done in our lab and our goal was previously looking at antimicrobial resistant genes, and those papers was already published. And also we did recently, we added long-read sequencing done by Dr. Ilya. And I am the first one in the lab actually doing the work with virus and developing the TS virus workflow, because the previous workflow was just for antimicrobial resistant genes called TEL-SIC, just to give you a background. So with that, we are able to overcome two main issues. The one is the sensitivity problem is overcome because we do probe capture enrichment method, and the second one is we don't need assemblies because we are able to have long rates. So with that, with the background and overview of the steps for the TS virus workflow, our first goal was to assess the efficacy of the workflow. And we did that by doing a head-to-head comparison of on-target versus TS virus library of each sample. So to do that, we used a total of 5 nasal swabs that were collected from weaned pigs, and we know 3 nasal swabs were qPCR positive for influenza, and 2 nasal swabs were qPCR negative for influenza. When do we submit those samples to the TS virus library, we observed that those samples that were qPCR positive for influenza, we obtained a on-target rate for influenza up to 87% compared to on-target libraries that we just got 0.7% of on-target rates for influenza. That was very interesting, and also another point that was interesting was to see that the qPCR negative samples, we actually didn't have any enrichment for those samples. Here are some plots regarding the coverage profiles for influenzas. So we plot HA and N and M1 and 2 genes for influenza. So in the columns is each gene. For influenza positive samples, for the 3 influenza positive samples that I had. So in the y-axis, you're gonna see the number of rates that align to influenza, and the x-axis is the position in the genome. The red line is the TS virus library, so samples that were submitted to the TS virus. And as you can see, we got not just a lot of on-target rates for influenza, but the coverage profiles were very good. We got very good horizontal and vertical depth of coverage. And for the blue line, which are the samples that were not submitted to the TS virus workflow, you can see we need to actually zoom the plot because the depth of coverage was very low compared to the samples that were submitted to the TS virus workflow. So the second goal was to investigate the TS virus limit of detection compared to the qPCR. And we did that by doing a dilution series. We used 3 serum samples that were PERS positive for qPCR and 3 nasal swabs that were influenza positive for PCR. So we did 9 serials dilution. We used 100 microliters of sample, 900 microliters of media. And then we test those samples by qPCR and TS virus. So the overall results, we are able to generate rates for all the dilutions for influenza and PRRS. We got a very good on-target rate for PRRS and influenza up to the dilution 4 and 6, which corresponds to a CT around 30. But one of the things that we observed is that as soon as the dilution get a little higher than 4 and 5, we observe lower on-target rates for influenza and PRRS. And this was typically less than 50%. And here are the graph. So we have A is influenza and B is PRRS. So in the y-axis, we have the percentage of on-target rates for influenza, and the same for B is for PRRS, and the x-axis is the CT value. So the colors are the dilutions. So the higher the dilution, you can see we have a lower on-target reads for PRRS and influenza when the dilution gets to number 6 to 9. So although we had the higher the dilution, we had a little bit lower on-target rates for influenza and PRRS. We can also, what we saw is that the coverage profiles for those higher dilution is still good, even though the on-target rate was not so higher. So here is a graph. I have the same, is similar to the other one. We have the y-axis, the number of aligned reads, and the x-axis is the position of the genome, and the colors means the dilution, so dilution 1 to dilution 9. And as you can see, the yellowish color are the highest dilution, and we have a lower number of on-target rates for PRRS in this case, but we still got a good coverage, horizontal and vertical coverage, even though we had lower on-target rate for PRRS and influenza in the higher dilutions. Here is a table just summarize what I just had. So as you can see, the higher the dilution, we got a little bit less on-target rates for PRRS and influenza. But in this case, for PRRS, we still got very good horizontal coverage and some good vertical coverage, which is depth of coverage depth. So our third goal then was to see, okay, we test using nasal swabs, serum samples, but what about more complex sample types? And our idea was to test the TS virus workflow using oral fluid samples. So oral fluid samples are currently being used in the swine industry a lot because these oral fluid samples, they are very easy to collect, and they are basically you put a cotton rope in a pen with a lot of pigs, and the pig's gonna chew on the cotton rope, and then after some time you are able to collect the liquid from the cotton rope, and that's what we call oral fluid sample. These oral fluid samples, they are very easy to collect, but they also are very challenged because they are very complex sample types. So we thought to use oral fluid samples and test those samples in the TS virus workflow, and for that we use 144 oral fluid samples that were already previous collect from a study that was performed from Dr. Jose Anglo in our lab and was already published. From those oral fluid samples, after they are submitted to the TS virus workflow, we are able to obtain a total of 65 virus across the 144 samples, and we had a mean viral richness of 14. We are able to detect some economic important virus for the swine industry that were detected in most of the samples, which is porcine astrovirus 2 and 4, porcine bocavirus, and porcine sapellovirus 1. Besides those viruses, we also were able to detect some other viruses that we have, that we already know we have economic importance for the swine industry, such as PD, rotavirus A, PRRS, which is porcine reproductive and respiratory syndrome virus, and influenza A virus. So we are not just able to detect those viruses in the oral fluid samples, but we also obtain a good horizontal and vertical coverage for those viruses. We also, so for those oral fluid samples, for this batch of 144 samples, we had data on qPCR, for PRRS for those samples. So we are able to do a comparison of the results that we obtain from the TS virus workflow compared to the qPCR data for PRRS for those 144 samples. And what we saw is that the TS virus workflow and qPCR, we had 35 samples that were positive, so they agree. And we also had 33 samples that agreed to be negative for TS virus and for qPCR. However, we did saw disagreement, no agreement between the qPCR and TS virus for some samples, a higher number of samples, 71. And we do think that can be because we also multiplex those samples we multiplex those samples when we sequence in 12 samples per flow cell. So we have some speculations about this number of disagreement between TS virus and qPCR. So the conclusion, we can detect multiple co-circulating viruses from complex samples using oral fluid samples, which is a very complex sample type. We can have an increase of on-target sequencing for the virus of interest using TS virus workflow, and this will lead us to allow us to do some genome analysis and variant calling for multiple viruses at once. The TS virus workflow is quick, has a quick turnaround time. It goes around 48 hours to go from raw sample to actually the data. We do have some opportunities, such as I just showed in the last slide, we have some less sensitivity for the qPCR in oral fluid samples. And as I said, we have some speculations, and can be that we have lower depth because we did multiplex those samples, 12 samples per flow cell. So we may increase we could increase the depth. We can also speculate that we have some competition from other viruses. So we can, for next step is optimization of the probe set. So we can actually design a different probe set targeting more specifically PRRS and all the other viruses that we found to see if that will change in the sensitivity for PRRS. Some things that we need to think about when you think about variant calling is that we still have some high error rate for the nanopore, even though right now we have the new chemistry. So for my project, we did use R9 chemistry. So we still have some high error rate, and we need some better algorithm to work with the bioinformatic results. The weird virus, so we did find a lot of virus that we still don't know if is pathogenic to pigs or not, and our question is, do they matter or not? So that's the opportunity to speculate right now. And for the challenge is the variability. So we still have some variability on the sequence depth per sample, the coverage profiles, and on target rate. And it's something that we also need to work on. But that's the next steps is to continue to analyze the oral fluid samples. So we have good metadata for this subset of 144 oral fluid samples. And our goal now is to do some modeling, so to use the metadata with production data such as mortality rate, biosecurity data, together with the data that we obtain from TS virus workflow to see if we can combine those. And also we're gonna keep validating more virus and sample types and we're gonna keep the optimization of the workflow. And when they say that is the optimization of the workflow in the lab, but also the bioinformatic workflow, because we do get a lot of data, and it's very hard bioinformatically to actually work with that. So we need to optimize the lab workflow, but also the bioinformatic pipeline. With that, I'd like to thank you for the funding, especially the Swine Health Information Center, all the lab members and the collaborators, especially Dr. Cristina Bolcher, Jonathan Bravo, my supervisors, Dr. Montes Remorel and Dr. Noel Nuez. With that, I would like to thank you for your attention. I'm open to questions, and also here is the Noel's lab in case you wanted to check or work. Yeah, thank you so much.
Questions?
Hi, I have a very ignorant question. I'm not a virologist. So you identified weird, I guess, unknown viruses. So I'm sure it's based on sequence. What about the sequence let you know that these were virus sequences?
Thank you for your question. So those weird viruses that I call weird virus, we already know they actually are in the pig. We know that pigs, they do have those viruses. But what I mean weird is because we still don't have actually a lot of data or knowledge about those viruses. So we don't have, for example, a lot of whole genome sequence about those viruses, or we don't know which role they play in the pathogenesis. Like, for example, they are there, but do they cause disease? They are together with the other pathogenic virus that we already know cause disease, so we still don't know. They are not very well studied, and I call them weird, but maybe it's not the best word. I could say that they are not very well studied virus. So that's the whole of one of the goals of the pipeline is to be able to detect those viruses, get a lot of genome characterize those viruses in a very well way, and then we are actually able to study better them to see if they are playing any role in the pathogenesis.
Thank you. That was very helpful. And this might also be a very ignorant question. You may well have touched on it. How did you get the acronym of TELS virus?
Oh.
Did you say that?
Yeah, it stands for, it's just how it stands for the target enrich long-read sequencing. So it's actually the combination for the two methods that we use. Here, target, enrich it, long-read sequencing of virus, because we do use the probes to do the targeting and then enrich for those viruses that we target, and then we sequence using long reads, which is a difference between the other platforms or workflows that have been done, which do short reads. Yeah.
Right, got it. Thanks so much.
Thank you.
I have a question from your mentor, who's not here. He asked me to ask you, in the specific case of PRRSV, which is so significant in the USA industry, were the genomes recovered enough to allow typing?
That's a great question, even though he's not here. So he asked if the PERS, we do have enough genomes that cover. So what we did for debates, we designed debates and we download all the full-length genomes for PERS that we have available. So what happens when we design the probes debate set, we don't have, we're gonna rely on how much data or whole genome sequence we have available in CBI. So for example, if you want a better a better representation of pairs or more accurate, what you can do is maybe to collaborate with a veterinary diagnostic lab, and they do have more sequence, whole genome sequence available for the sample, for the pairs variants that are circulating right now. And that's what actually we thought to do for the next probe set. But for this one that I showed the results, what we did, we download all the NCBI data, whole genome sequence available for pairs that we had at that moment, and for the other virus as well. So it may not represent exactly what actually we have right now, because of the limitation of the timing that we designed the probe. But the design of the probes are very, we can customize, so you can actually target the virus that you want to, and you can update the probe set as you want to. Yeah.
Excellent. Any other questions? Thank you very much. Thank you.